Hello everyone and welcome back to my channel Pharma Ki Pacha. Be a Pharma Pro. Today's topic what we are going to discuss is OS investigation. So let's get started. So let's begin. OS investigation. Full form of OS, out of specification. What is OOS? Results that fall outside the predetermined specification or established acceptance criteria set by the manufacturer or the laboratory. Example, if assay limit is 95 to 100%, results observed are 94%, then it is an OS. Let's go through the flow of OS investigation. If any OS results are observed, Analyst A informs immediate supervisor, preserving all test preparation. Next, the analyst and the immediate superior shall carry out a laboratory investigation as per the checklist. Is the OS due to laboratory error? If the answer is yes, correct the error and repeat the analysis on the same sample. This will be phase 1A. If the results are within specification, release the batch and simultaneously check the impact on the other batches. Is the OS due to laboratory error? If the answer is no, then hypothesis according to checklist shall be performed. This will be phase 1B. If the cause is identified during hypothesis study, repeat the analysis with the same analyst. If there is no cause identified during hypothesis studies, then carry out the investigation at manufacturing stage. This will be phase 2. Is there any assignable cause found? If yes, inform QA head and take appropriate corrective action. If no, perform analysis on the same sample by different analyst B. This will be phase 2B. If the results are within specification, if the answer is no, reject the batch. If the answer is yes, perform analysis on the same sample by different analyst A or C. If the results are within specification, if no, answer is no, then reject the batch. If the results are within the specification, the answer is yes, approve the batch. In detail in the flow of OS investigation. When you conduct quality control test on finished product, raw material or packing material and your test result falls outside approved specification, out of specification should be assigned and investigation should be initiated. Analyst has to inform his supervisor retaining all the test preparation. Supervisor shall check the results and fill the OS investigation form and assign the OS number from QA. Following are the OS investigation steps. First, Phase 1A, Preliminary Laboratory Investigation. Phase 1B, Laboratory Investigation Hypothesis. And Phase 2, Manufacturing and Laboratory Investigation. Phase 1A, Laboratory Investigation as per checklist done to identify obvious errors. Obvious error means direct or detectable evidences identified. Laboratory investigation checklist includes inadequate analyst training, incorrect instrumentation or equipment used, improper calibrated instrument or equipment, malfunctioning of equipment, instrumentation or equipment setup and operation not done properly, etc. Analyst error includes incorrect calculation, incorrect column used by analyst, incorrect standard or sample preparation and measurement errors, errors associated with dilution, sample spillage, incomplete transfer of sample, incorrect standards, incorrect samples or reagents or glassware used, instrument or material contamination before or during testing, etc. If any assignable cause is found during phase 1 laboratory investigation, then original results should be invalidated with justification and list should be repeated correcting the error. If no assignable cause identified during phase 1 investigation, then proceed with phase 1b investigation. Phase 1b investigation includes hypothesis study. Hypotheses regarding what might have happened should be tested. A study protocol describing the testing process is prepared and investigational testing is initiated with QA approval. Hypothesis study protocol must describe hypothesis for investigating the root cause, samples to be tested, execution process, process for evaluating data. If assignable cause found during hypothesis study, then invalidate the original results. Analysis should be performed by different analysts. If no assignable cause found in phase 1 laboratory investigation, then proceed for phase 2A manufacturing investigation. Phase 2A manufacturing investigation includes review of PMR, interview operator and supervisor, summary of aspects of manufacturing process that may have caused the problem, results of documentation review with actual assignment of probable cause, the results of a review made to determine if problem has occurred previously, and description of corrective action taken. If phase 2A investigation confirms OOS results, then batch should be rejected. An impact assessment should be carried out on earlier batches. If no root cause found in phase 2A investigation, then analysis should be performed on sample by different analyst B. This is phase 2B. If results are within the specification, analysis should be performed by different analyst A or C. If results are within the specification, then release the batch. If not, then reject the batch. Once the batch is rejected, further testing can be done to find out the root cause. Impact of OS on other batches should include ongoing stability studies, validated processes, test procedures should be mentioned along with corrective and preventive action, and conclusion should be mentioned. Thank you for watching my channel. For further more informative videos like this, please subscribe to my channel. Thank you.